Hi dear students, welcome back to one more session of genetics. Today we are continuing with DNA profiling, the part 3. When we do the STR profiling and when we use STR profiling in forensics, we can see that if you get two DNA profiles, one of a suspect and one from the evidence side, and if they are being found to be matching to each other, we say that the suspect was being found at the crime scene. And you are giving a solid evidence that the suspect's DNA and the evidence DNA is being found to be similar. So, but how strong is this particular evidence? Because we need to prove in the court that this particular profile is being found to be unique or it is being present only in that particular person. When this evidence is being provided to the court, of course, we should also give an implication that such a profile should be unique to that particular person. If the profile consists of a combination of traits that is being found to be extremely rare, the evidence becomes very strong that the suspect is the contributor. But if we find that the similar profile, DNA profile, can be found in different people, or it has been found to be a common thing in different individuals, then the significance of this particular evidence would be lost. So every time when you present a DNA profile like this as an evidence in the court, you also should have a probability stating that that is the probability of this profile to occur in different individuals. You should have a scientific estimation or a statistical estimation to say that this particular profile is being found to be. Now here we are looking 1, 2, 3, 4. 4 loci have been analyzed, and 4 loci have been analyzed, and for 4 loci you got different peaks. So the probability of getting a DNA profile like this in a population. Now if you're considering us, we are the Asian populations. So among the Asian population, the frequency of obtaining this particular profile is how much, that also we should estimate. And if the probability of attaining that particular profile is being found to be very less, or if it is very rare, then the evidence or the which has been provided, or the DNA profiling evidence which we have given here, is being found to be very unique, or it gives the strength of the evidence. So to in order to provide a genuineness for the DNA profile based evidence, we should also calculate the DNA profile probability. Now, what is it? Of course, we will be going to the details of it. This is how we calculate the DNA profile probability. It gives us the idea, or it gives us an idea about the probability that a match would occur by chance. So you check the allelic frequency, then you calculate the genomic frequency, and then you will say the profile sequence frequency is so much. That is 0.00014. That is the probability of this particular profile to occur in a population of individuals or in a group of individuals is being found to be very less. So if a person has got this particular profile, and if that profile is similarly found at the crime scene, it indicates that what happens, what it is, that profile is being found to be unique to that particular person, and it is not something which is commonly found among different individuals of the population which we are studying. So the strength of the evidence is mainly correlated with the when you provide also the DNA profile probability when we are dealing with DNA profiles. So in this particular session, we would be finding out how this DNA profile probability is being calculated, and what are the different terms which have been associated with it. So let's move on. As you are seeing now, these are the different loci which have been assessed, and these are the alleles. and how we are obtaining this, I will be explaining it. So that is the significance of it. So when you go to look into this, you can find we have to find out what is the allelic frequency, then you have to find out the genotypic frequency, and then you find the profile frequency. So let's move on and find out what are all these terms. Population genetics is, of course, the study which causes the patterns of genetic variations within and among populations. Now, if I am going to choose one person and find whether he is a culprit or not, of course, I want to check whether, like, I also need to check a population of people over that area also. So that's about, and the genetics of that particular population also has to be assessed. Now, what is allelic frequency? Allelic frequency is calculated directly by counting the number of alleles of one trait at a given loci and dividing it by the total number of alleles at the loci in a sampled population. And this is called the gene counting method. And if you go to consider genotypic frequency, it is calculated by dividing the number of individuals with one genotype by the total number of individuals in a sampled population. And each genotype at the loci can be calculated separately. And the summation of all the genotype frequencies at that particular loci should be equal to 1. Now, when I tell you like this, it's quite confusing, you might feel, but just look into this. If you have different alleles. Now, in this particular case, for CSFIP0, we have two alleles. Two alleles have been expressed over here. Similarly, there could be number of copies of alleles for A in a population. So that number of copies of these alleles divided by the total number of A and small letter a gene copies in the population will give a frequency of that particular allele. So we'll just take first a simple example before going into the human genetics. If you're going to take a population of P, you will have WW, capital letter WW, capital letter W, and small letter w as well as small letter WW. So the frequency of capital letter W, you just count how many alleles are there. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. The capital letter, there are 13. If this is a population, it's just for your understanding, I've taken a smaller population. So if you want to find out the frequency of the allele W, it is 13 divided by 18. What is 18? 18 is 13 plus 5 here. That is the total alleles which we have here is the total number of gene copies or the alleles which we have here is 18. So the frequency of the allele W, capital letter W, is 72%. And the frequency of the small letter w is 5 divided by 18 is 28%. And always the frequencies of all the alleles of a gene must added to be 100%. Now let's move on to the other one. The allelic frequency is also different from the genotypic frequency. What is the genotypic frequency? It refers to how often we see each allele combo. That is the thing. Now, if you have capital letter WW, the frequency at which capital letter WW is being found. So, 6 capital letter WW is there. So, there is a frequency of 0.67. Then the frequency of this particular genotype. So, genotypic frequency is different from allele frequency, and genotypic frequency, it implies how frequently that particular genotype is being found in a particular population. And of course, the phenotype is something which has been found to be a phenotypic character. How often a phenotypic character is being found to be found. So this is the example at which you have, so I guess you understood what is a allelic frequency. So when we are going to consider the forensic case, okay, we are doing this profiling. We are doing this profiling against different loci, and we need to find out what is the allelic frequency. Okay, the allelic frequency of each and every allele. Okay, Now, how will you find out the value of P and Q and all that? For that, you have to take a population of the individuals. Now, this is a table depicting the allelic frequency of 13. I've not depicted the whole thing, but I've just put a small part of it. So, if you're going to do a DNA profiling at the same time, you should also prepare a what? A frequency of that particular allele to occur in a particular population of individuals. So, when we are considering the DNA profiling of a particular population, and if we are finding out different loci, if you're doing the profiling of different loci, you need to find out the allelic frequency of that particular loci in that particular population. Now, if I'm dealing with African Americans, I have to prepare an allelic frequency of that particular loci. Now, if I'm going to take D3S1358, it can occur in different alleles like 12, 13, 14, 15, 17, 18, 19. So, comparing a number of people of African Americans, like now, I've taken 210 samples, and what is the frequency of that allele to occur in it? That should be calculated. At the same way, if you're working with Caucasians, you have to prepare allelic frequency like that, and if Hispanics, you have allelic frequency like that, or if you're working with Asians, you should have a allelic frequency of that particular population. So all the different loci, if you're considering profiling for 13 loci, the frequencies of different alleles for these 13 STRI loci should be calculated, and you need to prepare a table depicting these allelic frequencies. And using these allelic frequencies, later on we will calculate the what? See, in this figure we have see, where is it? Yeah, fine. Let me just one sec. Yeah, in this like I told you, the DNA profile probability is based upon the allelic frequency. So you have to prepare a database for your population, and you have to find out what is the allelic frequency of that particular individual. Now, this is a population. This is like 109 people were alleles were being observed, and the size of the database was 432, and they found out the frequency of that 0.25. So the 10th allele is being found have a frequency of about 0.25, and 11 has a frequency of about 0.31. So like that, when you're working with a particular population of individuals, you need to find out what is the frequency of different alleles of that particular population, and you need to record it as a what? You need to tabulate it and keep it in your system. Only after creating that database can you give the profile probability of that particular DNA profile which you are considering in your study. Now, once you get the profile, okay, the allelic frequency, you can also estimate the genotypic frequency. So I'm just moving from what allelic frequency, we are moving to the next one called the genotypic frequency. So you can, using the allelic frequency, you can also estimate the genotypic frequency, and this is mainly based upon the Hardy-Weinberg principle. So what is this? It is this principle states that you can allow the prediction of genotypic frequency based upon the allelic frequency. Now, the frequencies, and this can be calculated only if a equilibrium is being maintained. And what are the conditions that should be followed if you want to estimate the genotypic frequency? The thing is, the frequency of the alleles will not change from one generation to the next. That should be maintained. And the genotypic frequencies can be predicted by the allelic frequencies. And it is in such a way that the sum of the genotypic frequencies should be equal to 1. So P square and Q square are the genotypic frequencies of homozygotes, and 2PQ is the genotypic frequency of the heterozygotes. Now, if you have a person who is having two alleles, now P and P, so his genotypic frequency will be P square. That is the frequency of the P allele into the frequency of the second allele, that is p square. And if there's an individual who is having the genotype q square, okay, then the genotypic frequency will be q into q. The frequency of one allele into the next one becomes q square. And that is in the case of homozygotes. And if they are heterozygotes, the genotypic frequency is calculated with the formula 2pq. And according to the Hardy-Weinberg equilibrium, the sum of the genotypic frequencies should be equal to p square plus 2pq plus q square is equal to 1. So let's move on. So that's about the Hardy-Weinberg equilibrium. And we are again, I'm just continuing with the statistical analysis of the DNA profiling results as we had mentioned over here. So I told you, you have to find out the allelic frequencies. Now, once you calculate the allelic frequency, you can see here a person is having a profile with two alleles are there, 10 allele. He has an, consider he is having, you're checking this particular loci, that is C, S, F, and 1PO, 1P0. Okay, he is having a profile for two alleles are there for this particular loci, 10 and 12. So I will calculate the allelic frequency. Of course, if I'm working with a particular population, I should already have that one's allelic frequency, and comparing that, the frequency of the profile to have allele 10 is 0.25369 and of the allele 12 is 0.32519. So if you're calculating the 13 loci, you will calculate what is a what. If it is a, now here this is a heterozygote. So the frequency of that is 2pq. The genotypic frequency is 2pq. So the frequency of p into frequency of q, I got a genotypic frequency over here. Then I'm going to the next one, the next loci. That is also a, see, there are two different alleles, 14 and 15. So this is also a heterozygote. So the formula which we use here is 2pq. So this, the frequency of p and frequency of q is calculated, that into 2, I get a value over here. Now if I go to take the third loci, it is a homozygous, that is, he has only two alleles, 11 and 11. So the frequency is p square. So it is 0.168, that is 0.41 into approximately, 0.41 into 0.41, you get 0.168. And if you go to look here, D7 S3 S820, you can see he's having a heterozygote. So the formula 2pq is being used to find out the genotypic frequency. Like that, after getting a DNA profile, you will find out what are the alleles for 3 different loci. And of course, you have a database already regarding the allelic frequency in that particular population. And you calculate the genotypic frequencies. And once the individual genotypic frequencies for different loci have been found, the total profile probability is being calculated by calculating all of these, and then you express it. Now here in this case is 2.76 into 10 to the power of minus 14. This indicates that the probability to get this particular profile for these 13 different loci is 2.76 into 10 to the power of minus 14. That is, you must remember it is 0.000000 some value. So there is a very less probability of a person to have the same unique profile. So that means that profile which we have studied over here is being found to be very rare. So that's how it implies. And always when you provide a DNA profile in a court of justice, you should also calculate the DNA profile probability in that particular population of individuals which is there to make the evidence to be more genuine. So I guess you got a better understanding. I'll just get along. What is the significance of it? We do it mainly to find out the strength of the evidence which we are providing, and for that we need to provide the profile frequency, or the profile probability is to be calculated. And to calculate that, you need the allelic frequency and also you need the genotypic frequency, and you create the allelic frequency from a database based upon the individuals which you are dealing with. And if the allele of your sample, now in this case, it's like in this particular loci, you have two different alleles like 13 and 16. So the frequency of that, the allelic frequency of these two are individually calculated, and then you calculate the what? Genotypic frequency, and from the genotypic frequency you will ultimately calculate the profile frequency. See, the profile frequency is in this example is 0.00014. So the profile which we considered here has been found to be very unique, and the genuineness of the or the rareness of the profile will help us to conclude that the DNA profiling is a strong evidence. I hope you have gone through it and it's clear to you. Thank you for now.